Hello, I'm Vivian Cheung. I'm an investigator of the Howard Hughes Medical Institute, a professor of pediatric neurology at the Life Sciences Institute at the University of Michigan. Today in a three-part series, I'm going to talk about RNA variation from gene expression to RNA sequences. On our planet, there are tens of thousands species of plants to land and marine mammals. And we see this in nature from just the wide ranges of colors of trees and sizes and shapes of those trees. And in order, of course, all these biodiversity give us the beauty of nature, but at the same time, it's also fundamental to our survival because it provides us with the food and fuels that we need. In order for us to have this biodiversity, we need a lot of the building blocks. So we need at the molecular level a same extent of variation. What I'd like to talk about today is that variation in RNA. We know from the Genome Project that the sizes of genomes and the number of genes itself doesn't really give us all the answer to this complex set of functions. So on this slide, I show the number of genes in roundworms as well as in human. We see that the worms and humans basically have the same number of genes. And if we compare wheat to human, the genome sizes of wheat and the number of genes in wheat is actually much more than in human. So the complexity is probably not just from how big our genomes are or how many genes we have. We have known for a long time that it's not a straightforward one-to-one relationship between DNA, RNA, or protein. And what in fact we see is that one DNA sequence can be made into different forms of RNA transcript. And these different forms come from either alternative splicing or from RNA editing that actually changes the sequences of RNA from their corresponding DNA. And from these RNA transcripts, then different isoforms of proteins are made. So from one DNA sequence, we ended up having different forms of RNA transcript as well as different forms of proteins. So today, what I would like to discuss is mostly on RNA from variation in RNA at the gene expression level, as well as in RNA sequences. Oftentimes, geneticists really need this diversity in order to study the genetic basis of a trait. But oftentimes in mechanistic studies, in some way, this extensive variation can be viewed as almost as a nuisance. But what I hope to convey is that this variation is not an unwanted complication. In fact, it allows us to understand regulation. So let me start with talking about variation in RNA, in particular in gene expression levels. So in this experiment, what we did was a very simple experiment where we just measured the expression level of genes in B cells from blood samples. We collected blood samples from 50 unrelated individuals. Then we extracted B cells and measured the expression levels in those B cells. And shown here are examples of genes that showed very little variability across individuals. So such as in Parkin7 here, where individuals with the lowest and individual with the highest expression level, the range is very small. Whereas in the rest of the genes, as representative examples, we see a tremendous amount of variability. So on the y-axis is log2 scale of gene expression. Each individual is shown as a dot. So the individual with the lowest expression level, say, in this gene versus the individual with the highest expression level, there is at least 10 to 20-fold or even more differences. So if we want to know if there is a genetic basis to this variation, what I've shown you on this slide are data from unrelated individuals. If we want to know if there is a genetic basis, we need to look at people who are related, such as siblings in a family, or people who are genetically identical, such as in monozygotic twins. So we did exactly that. We measure, in addition to the unrelated individuals, we also measure gene expression levels in siblings and families, as well as in monozygotic twins. Here I plotted for you 5 examples where we plotted the variance across unrelated individuals in green, the variance across siblings in orange, and monozygotic twins in blue. In each case, what we see is that in unrelated individuals, there's a lot more variability than individuals who are identical, such as twins, suggesting that there is a genetic basis to this variation in gene expression. But we can look at this much more formally by calculating heritability. So in order for us to calculate heritability, we can ask, what does it really mean by a trait being genetically controlled? So if that's the case, then parents who given the trait to their children, or when the children inherited a trait from their parents, then they should be more similar to their parents. So here we're asking if expression level are the same thing, that if the children are more like their parents in terms of gene expression level. And so give you an example here for GSTM2, where on the y-axis are the expression level of individuals. On the horizontal axis is the average of their 2 parents. So we see individuals with low expression level come from parents who have also low expression level of GSTM2, whereas individuals who have high expression level of GSTM2 come from parents who have high expression level of GSTM2. So as a result, when we look at the slope of this graph, we see a slope of 0.75, which is rather high. And we see an example here also listed of genes that have high heritability. We're certainly not saying that for these genes, the expression level is completely genetically controlled, but they certainly suggest that there is a strong genetic component. So what does this really mean? We usually do not think of gene expression levels as a phenotype. But I think our analysis shows that it is very similar to the quantitative phenotypes that we're all familiar with, such as glucose level or blood pressure or size and shapes of fruits. So in that sense, that individuals' there are differences in individuals' height. Here, the expression level of genes differs across people, and there is a genetic basis to this variability. So this gives us an opportunity to take the expression levels and decompose them genetically. We can use different type of genetic mapping methods and ask, when we look at sites in the genome where people have differences in DNA sequences, whether there are some DNA sequences that allow us to decompose this range of gene expression. So for example here, there's some at a site where some individuals have DNA sequence that are CCs, some individual who have DNA sequence where they've gotten a C from dad, a G from mom, let's say. So they have a CG at that DNA sequence. And then some individual who have gotten 2 Gs from their parents, so they are GG. And as a result, we see here that individuals who have gotten 2 Gs from their parents have higher expression level, and people who have 2 Cs at that DNA site have lower expression level. And then if we go and ask, where is that DNA sequence, if it is close to the gene that we're measuring the expression level, then it suggests that that gene is cis-regulated. If that DNA sequence is on another chromosome, say in a transcription factor, we will have identified that the expression level is trans-regulated and identify the transcription factor that influence the expression level of that gene. So we did this systematically for all the genes whose expression level we're interested in. So let me walk you through what we do. We treat the expression level of each gene as a quantitative phenotype. So we identify families and measure the expression level from all members of those families. We then treat them as a quantitative phenotype and put them through first in a genetic linkage analysis. In these genetic linkage analysis, we're basically scanning throughout the whole genome and ask which of the chromosome regions are inherited at the same time with the trait of interest. So the way that we can track the different parts of the genome is to identify sites with the different DNA sequences. And this way it gives us markers or basically signposts to track different regions of the genome. And ask which part of the genome are often inherited with high or low expression level of a particular gene. And just to give you some details, the method that allows us to track both the chromosomal region as well as the phenotype is an algorithm developed by Hazelman and Elston. And together they are called the Hazelman-Elston algorithm. And basically, as I said, they search the markers in the genomes for ones that segregate, pass along in family with high or low expression level of genes. But mostly when we look at these large families using these linkage analysis methods, because there's only how often that chromosomes are swapped, that we ended up with a very large regulatory regions or very large linkage regions. So we somehow need to be able to verify those regions are indeed correct and narrow those regions. In order for us to narrow those regions, we carry out another type of analyses called association analysis. And this method, instead of just taking advantage of recombinations or segregations of chromosomal regions in family, it takes care of all the historical recombination, so we get much better resolution. So in order to do this, we do both at the population level as well as a family level using a method called the transmission disequilibrium test. In the TDT, we are able to improve the resolution of our mapping and get kind of narrow down the regulatory regions much more. So shown here are examples where over the 3,500 expression phenotypes we analyze, over 2,000 of them give us significant regions of linkage. So these are regions that are segregate with the expression level of genes, and on the y-axis are the p-values of how strongly those chromosomal regions segregate with the expression level of a particular gene. So for example, on the here, Chi3L2, the gene is on chromosome 1. By genetic linkage analysis, we identify the regulation is also somewhere on chromosome 1, actually very close to the gene itself, suggesting it's cis-regulated. Here's another example where the gene is on chromosome 18, but the regulatory region is on chromosome 3, so of course it's not regulated by something close to a gene, but something on chromosome 13. But you will see that the size of these linkage peak is very broad, basically covering the entire chromosome. That doesn't give us much information of where to look for the regulation. So we use linkage association analysis, tracking many millions of markers across the genome. Each marker, the result for each marker are shown in a black bar. And the markers that are most associated with the expression level of, in this case, IRF5, is shown by this marker, which is actually right over the IRF5 gene itself, suggesting that IRF5 is cis-regulated. But this way allows us to really pinpoint the regulatory regions, oftentimes to a nucleotide resolution. So overall, those 2,000 phenotypes that we looked at, about 20% of them, the linkage peak are very close to the gene itself, suggesting they are cis-regulated. The majority of them are somewhere else in the genome, suggesting those are trans-regulated. And we were very pleased with this 20/80 split, because if every gene is cis-regulated, we don't need to do all this exercise looking throughout the genome for the regulation. We can just look to the close to the gene itself. But if most of the genes are trans-regulated, using this genetic approach is very powerful, because we don't need to know in advance what mechanism it is. We can just search systematically throughout the genome for the regulation using both a combination of the linkage and association-based methods. Now let me give you one concrete example. We found through these genetic mapping that KDM4C is a master regulator regulator for the expression level of hundreds of genes. So what's KDM4C? KDM4C is the demethylase in the Jumanji family. In most of the time, histone marks regulate gene expression. And in this case, when histone 3 have 3 methyl groups, it acts as a repressive mark. It decreases the expression level of genes. So what this KDM4C does, it removes the histone, it removes methylation marks from the histones, therefore activating genes since it removed the repressive marks. So what we found was when we looked at the expression level of KDM4C, there's a tremendous amount of variability in the expression level of KDM4C. And when we looked at not only at the gene expression, but also at the protein expression of KDM4C, we see that individuals who have low KDM4C gene expression also have low protein expression level of KDM4C, and individuals who have high KDM4C gene expression also have high protein level of KDM4C. So we then carry out our linkage and association-based method and identify a set of sequence differences in the 3' UTR of KDM4C. So individuals with an A genotype of, at the 3' UTR of KDM4C have higher expression level of KDM4C, and individuals who have the G variant of, in the 3' UTR have lower expression level of KDM4C. So we know the KDM4C is cis-regulated. The KDM4C is on chromosome 9. So we look at the rest of our genetic mapping result and found that many of the linkage peaks actually are on chromosome 9 right over the KDM4C gene itself. So for example, here, one example is MAF2C. MAF2C is a gene on chromosome 6. And the linkage peak is mapping to chromosome 9. So suggesting that KDM4C is what regulates the expression level of MAF2C. And by association method, we were able to narrow and confirm that indeed MAF2C is regulated by KDM4C. So it's not only these 3 genes with linkage peak over chromosome 9. What we found was that there are over 390 genes that have linkage and association peaks mapping to KDM4C, suggesting that they are target genes of KDM4C. We then do experimentally validate that KDM4C indeed regulates the expression level of these genes by showing that KDM4C binds to the promoter of these genes and remove methyl groups from the histone 3 that's covering those target genes. So who are these target genes of KDM4C? They include many genes that activate cell growth, including well-known genes such as MYC, HRAS, and other genes that promote cell cycle progression, genes that regulate translation. So if that is indeed KDM4C regulates the expression level of these genes that influence cell growth, we should be able to see that individuals who have high KDM4C level versus individuals who have low KDM4C will have different growth rate, or at least their cells should have different growth rate. We measure growth by using 2 different assays. One with a simple assay, basically measuring how fast their cell grow using a growth curve. Another one is to use an assay called BRDU that basically allow us to measure how fast cells grow. So we see here that individuals with high KDM4C levels grow faster, both by a growth curve and also by the BRDU assays, compared to individuals who have low KDM4C levels. We then overexpress KDM4C, and by this BRDU assay, we see that individuals who have higher, once we have overexpressed KDM4C, those cells indeed grow faster by the BRDU assay. If KDM4C indeed regulates cell growth in diseases such as in cancer that's characterized by rapid cell growth, we should see higher KDM4C level. So we turned to 18 different types of cancers and look at KDM-C level between the cancer and the normal counterpart. We found that out of those 18 different types of cancers, 10 of them shows higher KDM4C in the cancer tissue compared to the normal tissues. We then asked if in these cancer tissue, if we knock down to KDM4C level, do we slow down the cell growth? So here's an example where we knock down KDM4C in colorectal cancer. First, we see that the target genes such as MYC following KDM4C knockdown have lower expression level, and in a consequence, the cells also grow slower. So here what we show you is that we started with a simple observation that KDM4C level varies across individuals. Then we asked whether we can decompose this range of KDM4C genetically, and we found that KDM4C level are cis-regulated by a sequence variants in the 3' UTR of KDM4C. We then identify a set of target genes that are regulated by KDM4C and work out the regulatory mechanism showing that KDM4C binds to their promoter and remove histone marks, remove methylation marks on those histones. And the high and low KDM4C level actually have a phenotypic effect on cell growth. So what I've shown you is that DNA sequence differences influence expression level of genes. And this is basically the essence of all genetic studies, is to identify sequence differences and ask which of those sequence differences affects disease susceptibility or phenotypic differences, whether it's gene expression level, cold color, or other type of phenotype differences. So genetic basically is to ask that what are the genetic blueprints that influence many of our phenotypes. So we're now familiar with the term it's in our DNA. What I like to kind of go one step further is to say that beyond our DNA, there are much information in our RNA sequences as well. So what do I mean by that? First, of course, there are many things that are beyond DNA. There are chromatin modifications that influence phenotype. There are ways that genes are alternatively spliced, so it ended up with different proteins, and that therefore one DNA sequence can end up having 2 transcript isoforms and then consequently 2 protein isoforms. But when I say that it's in our RNA, I really mean that it's in our RNA sequences, that there are RNA sequences that differ from our underlying DNA sequences. So perhaps this is not so surprising if we think about what is already known. The term RNA editing was coined by Robert Benne's group in 1980s, where they found that there are uridine insertions and deletions in the mitochondrial RNA of trypanosomes. And since then, and those RNA sequences were not encoded in the DNA. Since then, there have been other examples of RNA editing found in different organisms, including in human, where there are 2 forms of deaminases that remove amine groups from cytidine and resulting to uridine, and removing amine group from adenosine, resulting in inosines. Beyond these 2 types of deaminases, what our group have identified recently is that there are other ways in which the RNA sequences can differ from the underlying DNA. Since these are not driven by these deaminases, we just descriptively call these RNA-DNA sequence differences, or RDDs. I have already shown you that individuals differ in terms of gene expression levels. What I'm going to show you next is that editing levels also varies across individuals. For some, at the identical RNA editing site, some people have high editing level and some people have low editing levels. So shown here is such a graph. It's very similar to the graph I showed you before, except here what we show is plotted are the RNA editing or RDD levels. So on the y-axis is editing or RDD level. each individual shown as a black dot. So we show you here that there are some examples, for example, in the first column here, an A to G editing site in the gene, an elongation factor, where some individuals have low editing level, only about 40% of their elongation factor are in the G form, and some individuals, 100% of the elongation factor are in the G rather than the A form. And that's not for this one example only, but for a variety of examples, whether it's A to G editing or other type of RDD, we see quite a bit of individual differences. And just as in gene expression level, in individuals who are genetically identical, such as in twins, we see that the editing and RDD levels are much more similar to each other than, say, unrelated individuals. So what I've shown you in this section is that there's a lot of variation in RNA, and I describe in quite a bit of details how the extent of RNA, of gene expression level differences across individuals and how that is affecting cellular phenotypes. What I would talk about in the next two sessions is how RNA sequences differ across individuals and what are their phenotypic consequences.